Welcome back to the Human Life Expectancy channel. I'm your host, Tom Nodine. And with me again today is Dr. Jesse Poganik, instructor in medicine at Harvard Medical School. We recently posted a video on 3 of his recent studies that advanced the frontier of epigenetics. One was on TimeSeq, which presents a new way to process epigenetic tests that's up to 100-fold less expensive. Another one was integrative epigenetics and transcriptomics, which incorporates transcriptomic data, easy for me to say, transcriptomic data to produce and identify aging genes that could be used to improve predictors. And finally, a communication on what makes biological epigenetic clocks tick. So please check out that video on more for that. But in the meantime, we've received a lot of questions from our listeners, and today we'll answer those questions about health and aging and epigenetics. So welcome back, Jesse. You game for some questions?
Yeah, let's do it.
Let's go. So first, a few questions about aging. There continues to be a debate about what is actually driving aging and the mechanisms. What's your take? Are you a free radical guy or a stem cell exhaustion guy or something else?
Well, I'm probably an all of the above guy, to be honest. So,
Okay.
we wrote a paper a few years ago now, Vadim Gladyshev and I, explaining that, or making the case that, asking about the driver of aging or the definition of aging is almost asking the wrong question in some sense, because aging, of course, is many different things, right? So, stem cell exhaustion is a feature of aging. Free radicals and the associated damage, that's a feature of aging. Many other things are features of aging. But the question is, what is the underlying essence of aging, which is what we call this, underlying driving force in aging in that paper. And I think one of the motivations that I had for writing that paper was that this was shortly after I joined the field and had been thinking about things for a while. But in other fields, like in cancer, for instance, there are these very good explanatory models, like the hallmarks of cancer.
Yes.
And the reason I think that the hallmarks of cancer is a useful model is because you can link each hallmark to the underlying essence of cancer, which is mutations, right?
Yep.
Now we have the hallmarks of aging and we have the pillars of aging. But I would say that we don't have that underlying factor, the essence of aging. So what is my take? My take is that it is molecular damage accumulation, but I would say it is not anything as specific as free radical mediated damage. So there you can have damage at the level of DNA, you can have damage at the level of RNA, gene expression, any level of processing that goes into the journey between DNA and protein, damage at the level of metabolism, and then this molecular damage kind of builds on itself. And in the later manifestations of aging, you have the things that we colloquially associate with aging, like functional decline, like disease, and ultimately like death.
Yeah. And if I recall, back at the original Hallmarks of Aging paper was many years ago, it came out. Epigenetics was a key part of that. And it does seem like evidence has continued to show that epigenetics is a key part, if not the core cause of aging. Essentially, our cells kind of forgetting who they want to be over time, stop doing what they should do, starting doing some things they don't do, getting inflamed and all of that stuff. And suddenly we've got gray hair and all that. That's kind of what happens. So thank you. That's kind of the first question. Let's move on. Do you think methylation-based aging compares with other biomarkers of longevity, like telomere length or proteomic markers? How do you rank, if at all, the various biomarkers?
Yeah, so let me preface this by saying that in some sense, it's a little unfair to make this comparison. And the reason is because methylation has had, about a 10-year head start on other technologies. So you could maybe make a fair comparison between methylation and gene expression, which we touched on in the first video. Because those have been around for kind of comparable amounts of time. And as we said in the first video, the advantage of methylation over gene expression is that it's much more translatable between different cohorts. And gene expression is more subject to kind of technical artifacts that make the data handling a little trickier. There are ways to deal with this. People are working on it essentially, and I think that will change. So there are many different examples of biomarkers of aging, and with the Biomarkers of Aging Consortium, we're focusing mainly on the methylation biomarkers again because they have developed the most. But it's not to say that they're the best. Another thing we touched on in the first video is the difficulty of inferring biological information from DNA methylation data.
Yep.
So, this is a very valid criticism of methylation biomarkers. It's very difficult to understand the underlying mechanisms or even, what changes at particular CPG sites mean for downstream biology, like levels of gene expression, et cetera. But,
But that's why you're using transcriptomics to, in part, deal with that problem, right?
Yeah, or at least to get more, deeper insights, I would say. But, on the frontier, though, I would say that proteomics actually, even with the relatively few examples that have come out over the last, say, year and a half, actually the performance, depending on how you define performance, but in terms of things like prediction of aging associated outcomes, performance can actually be more robust in some cases for the proteomic biomarkers.
Cool.
And this may be related to the fact that the training, in some sense, is more robust because these have been done in the context of very large cohort studies, at scales that have not necessarily been done yet for methylation biomarkers. But yeah, beyond that, I mean, it's hard to, we could spend all day here talking about what people consider biomarkers of aging. So maybe we could just leave it there.
Okay, let's do. Other question. Here's one. Are there any surprising behaviors or exposures that significantly affect epigenetic age that the general public doesn't know about?
I think the general public, at least the people that are tuned into epigenetic or really any biomarkers of aging, I think they probably know, because the reality is that the most thoroughly validated interventions, if you can even call them that, are the things that are generally known to preserve health. So, healthy weight, avoiding alcohol, not smoking, good diet, exercise, avoiding stress, things like that. These are what have been robustly validated in terms of the epigenetic clock. So in terms of surprising activities, I'm not really, nothing comes to mind. But on the other hand, there've been a few interesting examples. So Steve Horvath has showed, for instance, that the level of carotenoids in the blood, which basically come from eating vegetables, this is associated with lower epigenetic age. So eat your vegetables. And then just another kind of silly example, I guess, is that there was a study showing that yoga reduces epigenetic age. So
vegetables and yoga are not silly. They are not silly. No.
But yeah, I think it goes back to this idea that I think what's known is what's already known. So
cool.
Use your best judgment of what you already know.
Some health-related questions. Here's one. Is there any way where your research has informed or affected your personal health practices?
Not really, because actually, I've done several of the tests, the clocks on myself, and actually, it's a kind of funny story. So the first time I did Grimage, I was like predicted as like maybe 3 or 4 years older than my actual age. And I didn't change anything in response to that. So I don't know.
I'm sure it was referring to mental maturity. I'm sure it was mental maturity.
Maybe. Yeah. I think it's also probably just that, academics don't have the best habits in terms of stress reduction and things like that. But anyway.
So you're not one of these people that takes 70 supplements a day to,
No.
Do you see a future where people get epigenetic age checkups as part of routine health exams?
Yeah. And I would say that there's an appetite for it, at least among clinicians that are tuned into this kind of longevity space, which is called by many different names, across medicine, anti-aging, healthy longevity, others. So I think that on the one hand, you have clinics that are doing this as kind of like their main bread and butter, and that's maybe a little questionable for the reasons we've discussed. But on the other hand, I would say you have very good clinicians with very high standards who are trying to integrate these tools into their practice, for the benefit of their patients. And I think the way most of them are thinking about that at this time is as essentially orthogonal readouts to the other kinds of profiling that they're doing already to monitor health and to try to ensure good health. So,
Exactly what we see in our insurance clients. They're doing the
right.
same thing. Mhm.
So I absolutely do see a future where these become, probably a routine part of clinical practice. And part of the reason is because there's an incredible richness to the data, of course, that you get. The number that we get from the clock is one number, but the reality is that the data that underlies that, um, is much more richly detailed than something you get from, for instance, a blood analyzer test.
Yeah. I
So,
mean, in the past, I've likened it to just after the telescope was invented. Suddenly you could see billions of things that you couldn't see before that are fundamental to nature. It would be odd if you couldn't discover some pretty
yeah,
dramatic things.
yeah, exactly.
And here we are. Now we're able to see the genome and the epigenome really for the first time, only in the last, let's call it 30 years.
Mhm. Yeah.
And so we can already see some amazing things, and there's a lot more to be discovered.
Mhm.
So, oh, here's a broad one.
Mhm.
Um, what impact could epigenetic engineering have on health?
On health, I don't know, because there is this still open question about how causal these epigenetic changes are to the aging process. So, from the research point of view, I think it would be really transformative to do.
Hmm.
For instance, you edit the epigenetic clock sites back to their youthful state and follow the phenotypes. Does the animal become younger, or does it reverse some phenotypes of aging, or which
Hmm.
phenotypes of aging can be reversed?
Yeah,
Things
you
like
see
this.
David Sinclair's, two mice next to each other, that
Exactly.
stuff. Yeah.
Yeah, and also, so I have a colleague, Ki-Jun Ying, who's he's a PhD student, but he's going to graduate next month, and he used a genetic approach called Mendelian randomization to try to sort out the clock sites between those that are causal, associated with mortality, and those that are adaptive, because of course we know that. As aging proceeds, right, there are the kind of damaging changes that happen that are believed to cause the changes that we see in aging that are generally bad. But the body has systems to address at least some of these, or at least for some time period, so that you have these adaptive systems that also increase as a function of age.
Hmm.
So it is actually a very important research question, and in the probably distant future, it will be an important question about, what can you do with this information for health? But the point is that you probably don't want to reverse every single change that's associated with aging, because you may be shutting off some of the beneficial adaptive changes that happen as a
Yeah.
function of age. So yeah, causality is hugely important. And I think, epigenetic editing will be a very important piece of getting to that question in
Cool.
the research
Cool.
context.
So, a few questions specifically with respect to epigenetic clocks.
Mhm.
Um, so, epigenetic clocks have come a long way in recent years. What are the most significant advances from your point of view?
Well, the latest kind of generation of clocks have attempted to do what we were talking about with the proteomic biomarkers, to try to
Hmm.
break down the signal into kind of subsignals, if you like. So Ki Junying, again, same graduate student, really really very talented, very prolific, also. So he had another study that I was a co-author on, where he basically decomposed epigenetic clocks into pathways. So he classified the different CPGs into, based on their genes and based on the pathways that those genes were associated with. He made clocks for different pathways, and so it's almost you can think about it as either a huge advantage or kind of nightmarish, that he can, the same data that you could produce a single clock like Grimage from, he can instantaneously produce hundreds of clocks for all these different pathways. So, but again, I think, it's a reflection of this underlying richness in the data that you're able to do these things. So it's, producing the single number that is associated with biological age or mortality risk. This is useful for obvious reasons. I think reasons that will be obvious to your viewers. But going further with the data that underlies those single numbers is, I think, where the exciting frontier is right now. And you see more and more examples of this coming out, I think.
Sounds great. And also that's on top of all the things that we discussed in our prior video around increasing the scale and reducing the cost using TimeSeek or some other similar approaches. Um, Another question, what are some of the biggest misconceptions about epigenetic clocks that need to be addressed? Anything come to mind?
Oh, that's a good one. Well, I mean, one thing is that I think that if people want to use these for themselves, they should understand the limitations. So, the companies that sell these tests often, the business model is sell the test, right? So
Mm-hmm.
they occasionally make what I would consider to be overstatements as to what the test can do, and what you can do in contrast, what you can do in response to getting a test result. So they're of course, the gold standard, as I'm sure everybody knows, is to do randomized clinical trials. And
Mm-hmm.
actually, this is another exciting frontier, I would say. So there was just a study out maybe 3 weeks ago from a large cohort in Switzerland studying the effects of omega-3 supplementation and showing that these reduced several different clocks that they tested in this cohort. So that was a really nice, robust study because it was a lot of people. The effect sizes were robust, and they showed effects over different clocks.
Mm-hmm.
So that would be, I think, kind of a gold standard way to test this. So then the question is, how does that translate to individual level action? And it's unclear, because on the one hand, not a lot of things have been, not a lot of interventions have been tested for their effects on epigenetic clocks. There are the issues of just kind of general validation that we've been talking about, the need for scaling these things to be able to get to that
Mm-hmm.
place where we're very comfortable with the validation and the validity of the clocks. Um, and then there's also the additional kind of wrinkle of we know from various studies, and I think just from kind of common sense and life experience, that people age in different ways, right? So even from the molecular level, there have been a lot of cool studies. For instance, from Mike Snyder's group, they coined this idea of ageotypes, and actually you see kind of a similar effect coming through in these latest proteomic studies, where certain people will have accelerated aging in particular organs or systems, for
Mm-hmm.
instance. So, what that means is that what I think that likely will pan out to mean is that when we try to intervene in aging, different people are likely going to require different interventions.
Yep.
And so, in the absence of really large studies where these things are tested and understood, it's very hard to tell because even if, even if a company has a small study that they've done with a handful of people and they show that a clock that they're selling is affected by a particular, intervention, it's not to say that you can reliably then say that I, as an
Yeah.
individual, if I take this intervention, my clock will be improved. And then that doesn't even go into the ultimate question, which is my health actually improved? Is my aging trajectory actually improved? So we need to connect these dots, I think,
Yeah.
to be able to do that. And so I think that was a really long, rambling way of saying that the clocks are great. They are transformational, revolutionary research tools. I think they're going to get there in the clinic eventually. But
Mm-hmm.
in the current landscape, if people want to individually test their biomarkers, they should understand the current limitations.
Absolutely. And we're working with insurers to do exactly that. I mean, we're
Mhm.
hoping to get, immense volumes of people trying different interventions so that we can actually see, for which people do interventions really cause meaningful reductions in biological age, or at least, slowing of the advance of age. or improvements in health. But it's going to take volume, and it's going to take time to get all that stuff done. Another question here about how you see multi-tissue versus single-tissue epigenetic clocks being used in clinical or insurance or OTC settings.
Yeah, it's a good question. So speaking from like the more clinical point of view, on the one hand, the advantage of the kind of multi-tissue approach, like you get with these. So I assume the question is, the idea of having a single biological age from a test or multiple biological ages. Is that the question, or is it more like
single tissue
one
versus
specific
multiple
clock versus
tissue?
multi-tissue?
Yeah.
Well, okay, if the question is single tissue clock versus multi-tissue clock, it probably depends on the context. So that's kind of a boring technical answer.
Mhm.
But the reality is, like for testing people, you're probably stuck with either blood or saliva. So um, it's probably less of a question than it is in the research context, where we can open a mouse and take essentially whatever tissue that we might want to take.
Yeah, take heart and lung.
Yeah, exactly. So
Yeah.
so in that case, I think if people are interested, my personal approach has actually been more towards the multi-tissue clock, and that's because we try to evaluate which ones we believe to be the most robust in each experiment. And the way we do that is that we take our control data where we don't expect to have any effects of whatever we're testing. We just expect to have the effect of chronological age.
Yep.
And we see which models are most robust at predicting chronological age in the control samples. And in several instances over the years now, maybe I would say even in all instances over the years now, it seems that the multi-tissue clocks are just more robust from a
Great.
research point of view. Now, on the other hand, with humans, as I said, you're stuck with blood and you're stuck with saliva. So most of the clocks that are out there are made for blood. And actually, those blood clocks, we also know from other research studies that those blood clocks actually tend to translate poorly to other tissues. So not necessarily saliva, interestingly enough, but for instance, we've done studies looking at things like liver transplantation and other, various other things where we have actual biopsy samples of solid organs. And the blood clock models actually translate poorly to those organs, which is probably not too surprising because they were trained in the blood. But yeah, so it really depends on the context of what you're trying to do. The future is bright and it's very exciting. And, I think we'll look back and be surprised that there was a time when we weren't more heavily intervening in aging.
Yeah. Well, we'll be here. We'll be here with you, hopefully. And Jesse, I really appreciate you answering these questions. Can we come back to you again as we get more questions?
Of course.
Great. And let's plot the course of, as epigenetics and our biological age capabilities get bigger and bigger.